Welcome to PK's classes. Today we will study about the structure of antibody. Antibody, also known as immunoglobulin. This is produced by our body in response to antigen. So antigen is a foreign substance which, when introduced into our body, our immune system produces antibody. So antibody protect our body from the antigens or the foreign substances. These antibodies help in eliminating the antigens from our body. There will be antigen-antibody reaction in which antibody will eliminate the antigen. And these antibodies are also called as immunoglobulins. And if we see the structure of this antibody, it is Y-shaped with 4 polypeptide chains, 2 heavy chains and 2 light chains. So this is the heavy chain, 2 heavy chains are there and 2 light chains are there. These 2 heavy chains are joined by disulfide bonds, 2 disulfide bonds, and heavy chain is joined to the light chain by means of a disulfide bond. Each heavy chain is joined to the light chain, respective light chain, through a disulfide bond. Then the light chain is divided into 2 regions. One is variable region and the other is constant region. And why we call it as the variable region? Because there is considerable variation in amino acid composition. There is considerable variation in amino acid composition in this area, and this is the area to which the antigen binds. Again, we can divide the antibody into 2 fragments, FAB fragment. This fragment is FAB fragment, and the other part, this part, is the FC fragment. And antigen binds to the FAB fragment, and FC fragment is the part which causes the biological activity mediation. Biological activity mediation means, suppose say some effector cells like macrophages. So the macrophages have receptors for this FC fragment, and we can call that FC receptor to which this binds. So let us see another structure of this one. So now what we have discussed, just we can have a relook. First, this is the structure of the antibody, which is Y-shaped. It consists of 2 heavy chains, 2 light chains, and the total structure can be divided into 2 parts, FAB and FC. So this is FC part, and then this is the FAB part. Then each light chain is divided into the variable region and the constant region, VLCL, and the heavy chain is divided into 4 parts, variable region, and the constant region is divided into 3 parts, CH1, CH2, CH3, constant, heavy, 3. Then if you see in the variable region, again we have hypervariable region, or also known as CDR, or complementarity determining region, complementarity complementarity determining complementarity determining region, CDR, to which the antigen binds, to this hypervariable region. Then, so if you see the CDR, there are 3 CDR from each of the heavy as well as light chain together form the antibody binding site, that the binding site to which the antigen binds. So this hypervariable region is present at the amine end of the antibody, the amine end of the the heavy chain as well as the light chain, and this end is the carboxyl end, COH end, and to this end the effector cells bind, because the effector cells like macrophages and natural killer cells, they have Fc receptor in them, and to which this Fc part binds, so that there will be the activation of the biological activity mediation. So you can see here another structure. So these are the hypervariable regions. Then there are sites like papain cleavage site, the pepsin cleavage site, the complement binding region to which complement binds, and the 2 heavy chains are joined to each other, and this region is called the hinge region. So this is the hinge region, we can see here, hinge region. And suppose this is the antigen. This antigen will bind to the antibody in the hypervariable region of the variable region, and we can call that region as the FV region. And the site of the antigen which binds to the antibody is called the epitope. So this is called epitope. So epitope is present in the antigen which binds to the antibody, and the site of the antibody which binds to the antigen is called as the paratope. So in actual there is interaction between the epitope and the paratope. Epitope is present in the antigen and paratope is present in the antibody. The light chain can be of 2 types, kappa and lambda, and heavy chain can be of 5 types, gamma, mu, alpha, epsilon and delta, and accordingly immunoglobulins are of 5 types. If there is gamma chain, it is immunoglobulin G. If there is mu chain, it is immunoglobulin M. If there is alpha chain, this is immunoglobulin A. If there is epsilon chain, this is immunoglobulin E. And if there is delta chain, it is immunoglobulin D. So this is all about the structure of an antibody. Thank you.